Hello, hi, namaskara. This is Kirtan and welcome to my channel Physiology at Once. Today I will be talking about the topic muscle proteins and sacrotubular system. Under this we will again talk about muscle proteins, sarcomere and sacrotubular system and its function. So let's start this video. Muscle proteins. They are of three types: contractile proteins, regulatory proteins and attachment proteins. Under the contractile proteins they are myosin and actin. And under the regulatory proteins they are troponin and tropomyosin, and in the attachment proteins they contain titin, nebulin, alpha-actinin, desmin, myomycin and dystrophin. Under this only titin, nebulin are important. So let's talk about the contractile proteins now. The first one is myosin. They mainly form the thick filaments of the muscle. The myosin filament is composed of two intertwined heavy chains, two regulatory light chains and two alkali light chains. They are again divided into three regions. Those are two globular heads, neck region and the long tail. So let's talk about these regions one by one. The first one is myosin head. The myosin head is made up of one heavy chain forming a complex with one regulatory and one alkali chain. So the two heads form two heavy chain, two regulatory and two alkaline chain. It contains two binding sites. The one is for actin and the other one is for ATP. So the alkali light chain stabilizes the myosin head and the regulatory light chain regulates the ATPase activity. So talking about the myosin tail, it lies along the axis of the filament. So it lies parallel to the filament. The myosin hinge or neck, it joins the tail to the globular head. It acts as a joint connection. So let's talk about the next contractile protein, actin. It forms the thin filament of the muscle. The actin filament or F filament or filament actin is made up of several globular molecules of G-actin. G means globular. The G-actin molecules wind around each other, forming a double-stranded alpha helical filament known as F-actin. So F-actin is filament actin, which is the main actin filament. Each monomer contains binding sites for myosin, tropomyosin, troponin I and other actin monomers. So the myosin actually contained two binding sites, which is one for actin and one for ATP. Actin contains binding site for myosin, tropomyosin, troponin I and other actin monomers. So talking about this, the F-actin undergoes a half turn every 7 G-actin monomers. So every G-actin monomers it turns and there is a groove formation. In this groove there is a tropomyosin molecule. So the tropomyosin runs in this groove. So let's talk about the regulatory proteins now. The regulatory proteins are tropomyosin and troponin. The tropomyosin, it is located in the groove between the two chains of actin as I discussed now. What is the function of it? Its function is to interfere with the binding of myosin head to the actin molecule. The next one is troponin. Troponin is again divided into troponin I, troponin T and troponin C. It inhibits the binding of actin. The troponin I inhibits the binding of actin to myosin by blocking the myosin binding site on actin. The troponin T binds the troponin complex to the tropomyosin. The troponin C, it binds the troponin molecule to the calcium. So you might have already read this topic in your 12th standard. So it's just like a revision for you. Let's talk about the attachment proteins now. The first one is titin. It provides framework for the thick filaments by connecting the Z line to the M line. The Z line is the main ending line of the sacromere and the M line is the middle line in the A band. It prevents the overextension of the thick filaments. Next one is nebulin. Its main function is to stabilize the length of actin filaments during muscle development. So the actin filaments are stabilized by nebulin. The other attachment proteins are alpha-actinin, desmin, dystrophin and myomycin. So let's talk about the sacromere in brief now. It contains I band, A band, H zone, Z line, M line, thin filament and thick filament. As you have already read this in your 12th standard, let's talk about this in brief. The A band contains the thick filament along with the thin filament. I band only contains the thin filament. H zone. It is the main zone which contains only the thick filament and it is in the middle. The Z line actually are the borders of the sacromere. The two Z lines form the sacromere unit. M line is the middle line in the H zone. So when the muscle contracts, the length of the sacromere decreases. Therefore, there is shortening of the I band, which is the thin filament band, but the length of the A band remained unchanged. A band is nothing but the thick filaments which also contain the thin filaments. So there is shortening of the I band, but the A band remains unchanged. So now let's talk about the sacrotubular system, which is the most important one and can be asked as a main question in the board examination. So what is sacrotubular system? They are the membranous structures surrounding the muscle fibrils. It consists of sacroplasmic reticulum and T tubules. So what are T tubules? They are also called as sacrotubules, as they are the tubular extensions of the sacrolemma. They are the extensions of the sacrolemma. They penetrate at the center of the muscle fiber and surround the myofibril at the junction of A and I bands. So they go inside the muscle fiber and they form a band in the junction of the A and I bands. The lumen of the T tubules contains the extracellular fluid through which the action potential is conducted over the surface of the muscle fiber and as well as the deep to the muscle or the interior of the muscle cell. So this is a nice diagram to understand the sacrotubular system. So this is the T tubule which is in the between, and this is the sacroplasmic reticulum. These two are the sacroplasmic reticulum at the sides of the T tubule. So these three form the triad and they are in the A and I band junction. Now talking about the sacroplasmic reticulum, it corresponds to the endoplasmic reticulum of the muscle cell. These are the slender tubules about 0.04 micrometer in diameter. The T tubules are 0.03 micrometer in diameter. They have an elongated position in the middle and dilated regions at both ends known as terminal cisterna. So these are the terminal cisterna around the T tubule and these are the elongated portion of the sacroplasmic reticulum which is covered in the A band region in the middle. So this is the A band region and this is the sacroplasmic reticulum which I am coloring now. So this terminal cisterna lies in close contact with the T tubules at the AI junction about which I talk now. This combination of the T tubule with the two neighboring cisternae is called as a triad, which plays a major role in linking membrane action potential to the muscle contraction process. So before talking about the process of sacrotubular system in the mechanism of the contraction, let's talk about the receptors and channel proteins in the sacrotubular system. The T tubule and cisternas are separated by a narrow gap known as junctional feet. So here in the triad region, between the T tubule and the terminal cisterna, there is a small gap which is known as the junctional feet in both the sides of the T tubule. So this junctional feet has two integral proteins, one in the T tubule membrane and other in the cisternal membrane. The T tubule membrane protein is a modified voltage sensitive calcium channel which is also known as dihydropyridine receptor. The cisternal side membrane protein is the ryanodine receptor. So this dihydropyridine receptor is a voltage sensor and this ryanodine receptor is a calcium release receptor. So there are also calcium ATPase present on the membrane of sacroplasmic reticulum. So now let's talk about the role of sacrotubular system in muscle contraction and relaxation. The T tubule action potential are linked with calcium release from the SR, that is sacroplasmic reticulums, that activates the contractile machinery. So when the calcium is pumped back into the sacroplasmic reticulum, the decrease in cytoplasmic calcium leads to the muscle relaxation. Thus the sacrotubular system maintains contraction and relaxation process by mediating calcium release and calcium uptake. So the calcium release and calcium uptake is the main process in this muscle contraction and relaxation. So the mechanism of calcium release. The depolarization of the T tubules induces the conformational change in the DHP receptors or dihydropyridine receptors or the T tubule membrane receptors. which further leads to the conformational change in the ryanodine receptor channel and thus opening it. So the conformational change in the DHP receptors further makes a conformational change in the ryanodine receptors. So the calcium is released and cross-bridge cycling is activated. The calcium is released by the ryanodine receptor channel. As I talked now, that ryanodine receptor channel is a calcium release channel. And the cross-bridge cycling is the main process of the muscle contraction, which I'll be talking in the next video. The further conformational change in the DHP receptor or dihydropyridine receptor allows the calcium in the cytoplasm, and this activates the ryanodine receptors, resulting in further calcium release. So it is a calcium-induced calcium release. The calcium in the cytoplasm again further makes the calcium release from the ryanodine receptors. So it's like a positive system. So now let's talk about the mechanism of calcium uptake. The membrane of SR or sacroplasmic reticulum contains sacroplasmic endoplasmic reticulum calcium ATPase. This is the main calcium ATPase that pumps free calcium ions from the sacroplasm to the sacroplasmic reticulum, from where they are moved to the storage site in the terminal system. So the calcium is uptake from the sacroplasm. So there is requirement of 1 ATP for relocating 2 calcium ions into the sacroplasmic reticulum. In the terminal cisternae, there is a calcium binding protein which is also known as chalcosterin. These chalcosterins favor storage of calcium at higher concentration. With decrease in the calcium concentration, the relaxation process begins. So it's all the game of calcium release and calcium uptake which helps in the contraction and relaxation process of the muscle. So talking about brief of the functions of the sacrotubular system, it transfers action potential from the surface of the muscle fiber to the interior of the muscle through the T tubules. It raises cytoplasmic calcium concentration by calcium release from the sacroplasmic reticulum. It ensures muscle relaxation by calcium reuptake by the sacroplasmic reticulum through the pump of sacroplasmic endoplasmic reticulum calcium ATPase. The system of SR or sacroplasmic reticulum acts as a storage sites for the calcium. That's it for today, guys. If you found this video helpful, please like, share and comment, and also don't forget to subscribe. Thank you.